Thank you. It's great to be here this morning after a 5-hour track and field meet last night till very wee hours. Though my son left with a silver medal in pentathlon, so all is good. Had to share the moments of a proud parent, right? Okay, so I'm going to talk a bit about, you'll note my title of my talk went from very generic, which I sent to everybody, to something more specific about clinical variant classification. And I spend time, a lot of time, some argue, does this work? No. Okay, I'll point. Is this supposed to work though? It's been a mixed bag. Okay. You'll just see where I'm gesticulating. So I spend a lot of time in the development of tools, standards, databases, other resources, both to inform our discovery in rare disease research, which I think Anne O'Donnell-Luria talked about yesterday, and also as I wear my hat as a clinical lab director and the chief genomics officer at Mass General to implement things in medical practice. So I'm not actually going to talk about the two sides of that, since Anne talked about our work on the left, and I'll save that for another time. But I am going to talk about a lot of the work we do in the middle because it influences how we think about bringing evidence, like mutational scanning work and functional data, into variant classification to inform both sides of this puzzle. So I'm going to talk a bit about ClinGen. Some of you are probably familiar with ClinGen. It's a now 10-year program that's funded by NHGRI, really with the goal of creating authoritative central resources that define the relevance of both genes and variants for use in medicine and research. It has 3 core grants, one of which I have here at Broad in partnership with Geisinger, and there's 2 others, core grants, and now we have a somatic cancer grant that Obia Malachi-Griffith lead. And then there's increasing numbers of disease-specific grants being funded through the different NIH institutes to cover their areas of work. And we really focus on aggregating genomic and health data in order to curate both gene disease validity, variant pathogenicity, dosage sensitivity in terms of loss of a gene or gain of a gene, and then clinical actionability. If I had a pathogenic variant in gene X, and I returned it to a patient, is there any action they could do to improve outcomes? So that's the work that we do, and then of course we disseminate it through our website and so on. There's a large number of people who are part of the leadership of too many organizations I help lead, ClinGen, and we now have over 2,800 volunteers that are part of expert panels from 66 countries across 123 expert panels. So it's an enormous engine now in terms of curation, with many more people than we actually can fund, which is more on the order of about 100 people. So a lot of work, and I encourage you to get involved if you're not already involved, because we bring scientists who understand functional assays into our fold as well. I want to talk a little bit about the curation landscape, because I think about this in 2 ways. On the right is the expert curation we do, but we also rely on people sharing primary evidence in their own work. And in the form of variants, those get submitted to ClinVar, which is a database of variant classifications. And now we've launched the GenCC database, which is a database that anyone can submit gene disease validity classification. And I'll show a little bit about that in a second. So before we can actually get into variant classification, you have to understand the evidence that the gene is even involved in disease, because you can't call a variant pathogenic, which means causal for disease, if you haven't implicated that gene first in disease. So we really are trying to curate all the gene-disease relationships that have been published, and we developed a framework that was published now a number of years ago, and it's semi-quantitative. There is still professional judgment involved in evaluating the validity of evidence, but we, as much as possible, try to do it in a standardized, point-based, quantitative way, looking at both genetic evidence, things like case control cohort studies or family segregation, as well as experimental evidence, model organisms, in vitro assays, all sorts of evidence. And then based on the number of points that are scored, you get one of these categories of definitive, strong, moderate, and so forth. We then worked with American College of Medical Genetics to sort of turn that framework into recommendations for which genes should be included in disease panels for clinical testing, with the idea that moderate and above should be in a disease-focused panel test, and limit and below be left for more exploratory or at least exome genome sequencing level. And this is just a look at the framework with the different points. It's the same between strong and definitive in terms of points, but definitive really means it's been replicated and stood the test of time for at least 3 years before it gets to that level. So to date, ClinGen expert panels have curated 2,632 different gene-disease relationships. And you can go to our website, and all these fun little circles are on there. And up in the right, I copied the sort of outcome of those, which is about 23% of gene-disease relationships that have been published have insufficient evidence to support the claim. And I can tell you, having been a part of curation of these genes for many years, the ones that are limited and below almost never move up. So, there's a few that had an interesting case just come out and it's in some supplement, and sometimes they move up, but almost never. They're usually just really insufficient evidence ever to begin with, never to be moved again. So really buyer beware when you're reading publications. I mentioned the GenCC database that we launched now just a couple years ago as a place to, well, the original intent was to harmonize some of the existing resources in terms of how they talk about gene-disease relationships like OMIM and Orphanet and Genomics England Panel App. But then we discovered all these clinical labs are also curating gene-disease validity for forming their multi-gene panels. And none of that data was public. So we really created an environment to bring all of this stuff together. I like to call it the ClinVar for genes. But there's a whole steering committee of representatives, all the groups that are submitting their data into this. I'm part of the Greger Consortium that Anne probably mentioned yesterday for rare disease discovery. We're also now trying to encourage the place to share early stage candidate genes as well, albeit marked with that limited or below evidence, but as a way to more publicly share these early stage, link to preprints, things like that, or even just summarize your unpublished data. So we'll be soon submitting all of our candidates for the Greger Consortium from our work, as well as the ones that are more established, into this database. And Marina DiStefano co-chairs GenCC with me, although she does most of the work. This is just a screenshot looking at the GenCC database website, where you can see different groups that have submitted for GGB3 and their strengths of evidence for each disease that they're claiming. And by bringing all this data together, harmonizing it, we can then compare, identify discrepancies. And that little panel on the right is our work to now resolve any differences between the submitters. And so we have monthly calls to go over anything that's discrepant, and we gradually resolve those differences. So this has been a great effort. And I think the most startling work was in our Brugada syndrome expert panel, where of the 21 genes implicated in that disease, the experts felt that only 20 of them had sufficient evidence and disputed, only one of them, and disputed the other 20. So, and I think, although this figure is actually from our cardiomyopathy group, I think one of the challenges that we see in the field is sometimes, so what this is showing you is the scoring evidence for each of the genes, with red being genetic evidence, like observations in patients, segregation, de novo occurrence, versus the blue being experimental assay evidence, functional studies. And you'll see a lot of these genes that are in the limited category are mostly functional data. And for certain assays, they may not recapitulate human disease. And that's where sometimes things go wrong. And I think this is also what happened with the, probably with the Brugada syndrome, where equating just dysfunction of an ion channel does not necessarily mean that will lead to a true arrhythmia phenotype. So really thinking about functional assays and whether they accurately recapitulate human disease is a critical step in this effort. Okay, so that's my brief foray into the gene disease part of it. But since I know a lot of you are very interested in variants and their functional effect, we're going to spend the rest of the time really thinking about variant pathogenicity. And I like to think about this in my sort of four-pronged approach to curating the world of variant classifications. And while For gene-disease relationships, I am confident that ClinGen will eventually get through all of them. There is no way ClinGen will curate billions of variants in the human genome, right? So we have to take a different approach. The first is, can we put common standards out so that everybody out there who's curating variants is actually doing it with some similar approaches and consistency? So we'll talk a little bit, in fact, we'll talk about a fair bit about both the sort of standards that we've developed and are now soon to release the next phase of. But just because you have standards does not mean everybody implements them the same way. And in fact, public sharing of variant classifications is really critical because not everybody finds the same evidence, not everybody interprets the evidence the same way, and being able to transparently see how someone has classified a variant and the rationale, when they include their rationale, is useful, is really important. And then we can quickly identify where there's discrepancy and curate and sort of focus our expert panels at the bottom on those variants that are discrepant between submitters. And also, sometimes we focus on ones where everybody says it's VUS, but there's lots of submissions, which means there's actually probably a lot of evidence out there. And if we bring all those groups together and share all that evidence, we might be able to move those variants up or down. So those are the areas where we focus our expert panel efforts, even though there's no way they'll ever classify all the variants. And then also equally important, particularly for this group, is we need the ability to access a shared evidence base. You know, I put NOMAD up there. We released data through NOMAD. MaveDB, also a great example of really starting to help share the functional evidence base. If we have time, I'll mention a bit around some of the work in Global Alliance, expanding our matchmaker exchange and beacon network efforts. All right, so that's sort of a big picture, how I think about the world of variant classification. But coming back to the standards, so most of you are probably familiar that 2015 we issued what wasn't the first version, but probably the first version that really thought about how to think about evidence in this through ACMG and AMP. And even for the first time defined those 5 terms. The only one we had in common use was uncertain significance. The others were all over the map. So we put those standards out, but then we immediately formed the ClinGen Sequence Variant Interpretation Working Group. And the goal of that was to refine and evolve those guidelines as they were tested and deployed by our community. Also, we have all these expert panels developing disease and gene-specific specifications of these rules, and we needed to review what they were doing and harmonize their efforts across our expert panels. And then really an ultimate goal is to move this framework towards a more quantitative framework. And I will say, when we were developing the original ACMG AMP 2015 standards, we actually had a points-based system. I had converted the whole thing to points, and we were going to launch that, but in the end, the committee said, if there's very specific point values for every variant, people are going to think we understand more about this than we do. And there was actually concern that we were going to overemphasize the accuracy of our approaches and how we were doing this, which was unfortunately still largely based on professional opinion in a lot of ways. So we retracted that and went back to this sort of strong, moderate, supporting levels to sort of say, we're still not sure how this whole thing works. So that was sort of the limitation of where we were at back in 2015. Now, since then, this is a screenshot of the sequence variant interpretation website from ClinGen. And you'll see on the left, there's all sorts of different guidance around more specific guidance for applying each of the codes that are in that original framework. Some of those are publications, some of them are white papers, but really helping the community sort of think about how to apply these evidence, because there was huge variation, I can say, in how they were being applied. And much of this work is now being incorporated into the newest version of the guideline. All of the disease-specific specifications from our expert panels are on a separate website called the Criteria Specification Registry, and so you can go there and you can either look up by code. I want to see how every group has specified the functional code, or I want to see how the hypertrophic cardiomyopathy group has specified all their criteria. So they're all in there. To date, we have, just looking this morning, 166 gene disease specifications spanning 155 genes. So that's great. That's a lot of work. On the other hand, there's a lot of genes yet to go. And maybe if we have time at the end, that we can talk about some of the relative priorities of our expert panels and how we think we should be directing them. We also have some guidance on dealing with those very annoying low penetrance variants that are both in Mendelian genes but happen to be hypomorphic alleles and other things like that, as well as sort of dealing with some of the very low penetrance, really non-monogenic disorders. And so we have frameworks now thinking about those genes. But now I'm going to get back to the current evolution of the sequence variant classification guidelines. So that 2015 version that I've been talking about was actually the third version from ACMG. And so we've now retrospectively numbered them, even though they weren't numbered before. And so we're calling the newest one V4.0, which also implies that it is our intent to more keep the committee together and more regularly update with iterations, as opposed to doing one, throwing it over the fence to ClinGen, letting ClinGen iterate, and then trying to regroup at the end. So now ClinGen is a part of this guideline, and we'll be releasing iterations on a regular basis. We've also been trying to work with the copy number variant group, and there's been some sort of back and forth, but they've kind of been off cycle. The CNV group has now reconvened. They're now taking the Bayesian framework we're using in SV 4.0 and applying that. So, and eventually, we'll probably have a single guideline, just not quite there yet. So this is the current committee that's working on, I keep changing the year that we're labeling it, but it now spans ACMG, AMP, CAP, and ClinGen, co-led by Stephen Harrison and Les Biesecker. And we've been working hard for like the last over 2 years. So some of the limitations of the current guideline, a lot of subjectivity in applying the evidence criteria, as well as ambiguity in the strength of certain criteria. A lot of lack of clarity around combining evidence and double counting certain evidence in multiple codes. Lack of guidance of when you have both benign and pathogenic. Some people read the guideline and say, well, then everything has to be VUS if you have anything on either side. I'm like, no, that is not what we meant. And then there was certain criteria that really were just inappropriate and highly misused. So really, PM2 absence in the population should not be any evidence. Most of variation is unique to individuals, or a lot of it. It's not evidence of pathogenicity, so we're getting rid of that. But the way the framework was built kind of depended on multiple lines of criteria to get anything. And so we couldn't strip it away immediately. And so we left it at the supporting level just to have the framework be able to deal with it. But when the new framework comes out, that will be gone. Then this odd PP5, BP6 reputable source we threw in there because you may recall Myriad was the monopoly on breast cancer testing because of patents before they were ruled unable to be used. And so we had this issue that they had millions of data not shared that underlies some very good classifications and how do you use that sort of stuff. So we had this reputable source. That is the most poorly applied rule to date. We've told everyone you can't use it anymore, but they still do. With things like, it's in ClinVar as pathogenic. Oh, okay, I'll stop harping. Anyway, moving on. So we are now moving to a Bayesian framework. This has been published. John Teptesian led that work and sort of reformulating all of the evidence levels in a Bayesian framework. Now we've converted that to a point-based adaptation, so it's easier for labs to apply specific point values. So that's kind of underlying the whole thing. So each of the different levels has a certain number of points that correlates to it. And this is all draft. It is not finalized, so things may change. But we're really creating new codes, so it'll be crystal clear when somebody's used the new guideline, because all the code labels will be changed. There'll be an area for thinking about human observational data. We've actually put out recent paper thinking about locus specificity, how you use phenotype and segregation data, with really the fact that both are the same concept. So if you have segregation, you are sort of pointing to a specific locus by virtue of meiotic segregation evidence, narrowing down the genome based on meiotic segregation. But phenotype specificity actually does the same thing through a different type of evidence. The more specific that phenotype is to a specific locus, actually just narrows down to a specific gene, right? And if you have such a unique phenotype that actually implicates a single gene, so that you have 100% likelihood that that gene, because of a unique phenotype, then adding segregation data gets you no further evidence. So you can kind of think about that the two are linked. So we're actually now in the new framework kind of linking them and also better specifying how to use phenotype using diagnostic yield. So if my patient has a phenylalanine level above X, and I test them for the PKU gene, what's the percent likelihood I will get a pathogenic variant? If it's really high, like 90%, that tells you that phenotype is highly specific, and you can add high point value. On the other hand, if you bring your autism patient up and test them, what's the yield on an autism patient? Well below 5%, because there's really no specificity of that phenotype for any one gene, so you get no points for phenotype. So that's just an example of how to think about the phenotype specificity. As we all better understand linkage and segregation, but that phenotype can be equally implicating a single or small number of loci. We've also built in the idea of ruling out other loci. So if you do a panel, and it's highly specific for a set of genes, but you rule out variation in all of the others, it helps you develop evidence for the remaining gene or the gene you have a variant in. So those are some concepts we've further developed in the new framework. And then we have this whole section of variant impact. It's all part of one framework, so you can prevent the double counting issue of various codes that we had. And then the functional effect at the bottom, which I'll dive in a little bit. But I'm going to give you, since I know a lot of you like to work on missense variants. So this is a little, when people see these decision trees, they're like, oh my God. Yeah, they're a little detailed, and I'm giving you the bird's eye view of these. But at a high level, what's happening here is you start out with a missense variant, and then we're telling them you need to look at two concepts. One, the impact of the protein amino acid change. But then every time you have a missense variant, you need to also think about the splice impact. And that's not consistently done, I can tell you, in labs. So the framework now forces you to look at both and then use whichever path gives you the higher score, right? And that's really important concept. So now if we dive into the top part of this at a level you can actually see something, this is sort of zooming in on the three critical parts of that decision workflow. So the first is a lot more guidance around the use of in silico predictors. And some of you may have seen the paper from the ClinGen SVI in silico working group that really validated a lot of the predictors and developed a quantitative framework for them. And so we're using that directly. They're now working on evaluating other predictors that are coming out, alpha missense, Eve, other things like that. But it gives you a quantitative output of those scores based on that validation. Then, now in every one of these decision trees, we now have, you've got to look at the transcript. And so the current is a bit generic of, if the residue is in all transcripts, fine, you can proceed. But if it's in most of them, but not all, your whole score gets immediately cut in half. And then if it's not in the clinically relevant transcripts, it's nothing. That's a little bit non-granular, but that is where our expert panels are really going to come in, really look at those transcripts and give much more guidance. And all of our expert panels do that. Like this exon in BRCA1, those two exons that if you have loss of function, do nothing, they will specify what to do with different exons. So we're kind of leaving it there in less granular, but that's where work will need to be done. And then on all the decision trees, there's the comparator variants, where you can add some extra points because there's already a known pathogenic missense variant, different nucleotide, but the same amino acid effect. And by going into the splice section, you will deal with that fact that sometimes there's what appears to be the same amino acid, but it's operating through splice, will be dealt with down below. But here, you'll get points for that different nucleotide, same amino acid, versus different amino acid, but in silico predictor predicts at least the same or above effect. And then on the benign side, there's a benign variant at that same site, those kinds of, and so that will add or subtract points to your thing. And then that will give you a final score at the end, which is capped at 8 points for missense sort of framework. But you also, and I don't have time to go through every framework here, but there's the whole splice prediction framework, that will give you between -4 and 8, and then you compare them and you take the higher of the two scores. Is there any possibility that both things are going on? Yes, but every time we have some discussion going into the weeds of that scenario, we say, leave it for 4.1 and move on. So yes, there's all sorts of biological complexity, but most variants do probably act through one major mechanism, and so we're kind of guiding you to focus on that. So this side, where you can't see much of anything, but is the functional frameworks. And they span animal models, in vitro assays, and actual splicing cDNA assays. And so just to give you a little bit of a flavor, the top there is a knock-in variant model. So if it's a knockout, it's not actually specific to the variant. It informs how we use the PVS1, or what will be newly labeled kind of loss of function code. But if it's a knock-in of your specific variant, then you have to look at things like how specific that phenotype is for the disease in your animal model, and other things like that, and the matching inheritance pattern that determines the score. If it's an in vitro assay, we're actually using the Bernick et al. Framework that was published by ClinGen to inform that, and really trying to get a quantitative output. So if your actual functional assay, you can do an odds path assessment of the likelihood of pathogenicity with your assay, then we can use that and directly assign the point values based on the odds of pathogenicity, and that will actually allow you to get a higher point value. In contrast, the Bernick et al. Is more about like how many pathogenic and benign variants were used to validate it, and that will give you some number of points, but it's actually lower than if you do the full odds of pathogenicity evaluation for your assay yourself. So, and obviously this is where joining expert panels and really thoughtfully thinking about this from an expert's perspective, where you understand how well those functional assays really recapitulate the function of those proteins and the pathogenicity and pathogenic mechanisms of disease, are critical to understand how to score these evidence. But in the end, you will get some point value to contribute. So just to give you some example of this, this is our hearing loss expert panel that's been around for quite some time. So we developed the class of, all of our expert panels first define their group, that gets approved, their scope of work, then they develop their classification rules, the specifications of how each of these rules will be applied. They pilot those, and they actually then get reviewed by our SVI VSEP review committee. And then once approved, they can pilot them. They then do the pilot, then they turn in the results of their pilot, any changes they may have made to their rules, and then they get final approval. So it's a 4-step process that also allows us to get FDA recognition for the work of these ClinGen expert panels. So our hearing loss group did that, and this is just an example of using the Bernick et al. Functional framework for a particular assay, in this case for GJB2, the most common cause of childhood hearing loss, and the Xenopus oocyte electrical coupling assay for gap junction function. So these were all the papers published on that particular method. The number of pathogenic and benign variants used to test those assays. In all, it ended up 23 likely path and 4 likely benign and benign. So based on the Bernick et al., that led to a strength of moderate for this particular assay and use in variant classification. So what I'm now going to show you is how our expert panel originally classified a particular variant here, and then how the new framework is going to lead to its classification, which is actually different. So the original is summarized, written out, but the 2 codes that were applied were PS3 moderate, based on that functional assay, and then PM3. PM3 is the case observations, and this is a recessive disorder, so the variant was found in compound heterozygosity with another pathogenic variant. That is good evidence, too. So for the sum of evidence up there, so reminder, no more points from absence and nomad. That's fine. But here's those. So there were 1.5 points per case for the 3 cases of finding compound heterozygosity with other pathogenic. Now, they didn't phase them, so they don't get quite as many points in the paper. The Rebel score was actually not great, so that scored no points. But that functional assay, based on that moderate using the Bernick et al. Framework, got 2 points. In the end, that's 6.5 points, which actually at the 6 to 9 is likely pathogenic in the new framework, whereas the original one was a variant of uncertain significance. Now, interestingly, if you look in ClinVar, 3/4 of the labs all say likely pathogenic. Why? Because any person who's been around long enough knows that this level of evidence is probably likely pathogenic. Like, that's my professional opinion. But if you formally follow the 2015 framework, it was hard to get some of these categories of evidence high enough to actually score them to pathogenic if you didn't use the crutch of the PM2 and the various non-specific double counting codes. So what does some of the labs do, and I'm guilty of this, is you kind of finesse a few other codes in there to get what you think is the answer. And that's not a good approach, but that's probably why those labs all are saying likely path. But the expert panel using the codes exactly how they were defined and how FDA recognized didn't get you to likely pathogenic for this scenario, when it really should have. So in the new framework, we're actually allowing the good evidence to get higher points and not relying on the bad evidence to get us there. So just some glimpse into where the new framework is sort of taking us. So I will also point out a significant change that's about to happen, which is we're going to let variants with less evidence get into the benign, likely benign category. So now a -1 point gets you right to likely benign. That was not true of the original framework. And in part because in the original framework, the mind of most clinical lab directors used to doing fairly targeted clinical sequencing was you start out and any variant is uncertain until evidence sends you either way. We're now in the genomic world. Honestly, if I do a sequencing test, most variants, prior probability is that it's benign to start with, right? If you really think about it, unless you were testing a gene highly specific for the phenotype. So if I have a patient with cystic fibrosis, and I look at the CFTR gene and there's a variant there, yeah, I would start with VUS, and go from there. Why? Because the phenotype correlation. But I just explained to you that phenotype score where you can get a fairly significant number of points for how specific that phenotype could be by that alone. So, but in the absence of scoring for phenotype because of a very close match, and if you get rid of that, and that's not the scenario you're starting with, then any variant probably meets the definition of likely benign, which is 90% likelihood that it's not disease-causing. So we're really trying to change the thinking. And if there is zero evidence, and zero evidence also means no in silico predictors saying it's pathogenic either. So those predictors have gotten better over the years. But in that scenario, those variants are actually, a lot of them, going to hit likely benign. And I'll actually show you some data. The other major change that's happening is the subdivisions of VUS. And in a bit of the rest of my talk, I'm going to talk about why that's so important. But I'm going to show you a little data I got just in the last week. It's unpublished, but Hila Aziz, who's on our Hearing Loss expert panel, gave me permission to show this. So because we've been road showing some of the stages of the work we're doing to get feedback from the community, she took the draft version of the V4 and applied it to 179 missense variants that were all VUS. And this is what resulted. So 49% of them became benign, likely benign. That's exactly based on what I just told you about sort of a reframing of the framework. 3% actually moved to likely pathogenic, and 6% became VUS high. Again, I'm going to talk a little bit about that, but this gives you some sense of kind of how this evolves to the new world. All right, now a little clinical sideline. So this is a lawsuit that happened over a few years ago, which was a lab was sued for misclassifying a variant. And so this was actually Quest Athena lawsuit. Here's the story. Doctor orders a genetic test. There's a VUS put on the report. In the report, it says you should test the parents to look for de novo occurrence. Physician does nothing with this test, does not test parents. In fact, doesn't even give the copy of the report to the patient. And off goes the family with no really information. That VUS was in a gene for Dravet syndrome, form of epilepsy. That gene is counterindicated for certain drugs treating epilepsy. So they had no awareness that this gene, this VUS, had been found. Child was treated with that drug and died, largely because of that treatment and the lack of information about the genetic basis. Now, it was a VUS, albeit, so largely most general recommendations are not to necessarily care for a patient based on a VUS, but it might indicate some things you should do, right? And they didn't do that. So the family actually didn't even know the test was run, never had the results. But it wasn't until they were planning their second child that they went digging to try to understand what happened with their first child, why they died. And they came across the medical record with this report. They tried to sue the physician, but because of the time lapse and statute of limitations in South Carolina, couldn't sue the physician, so they sued the lab. The lab wasn't really at fault. They had actually interpreted the variant okay and suggested de novo assessment with parental testing, which never happened. So that's a bad story. We want to avoid that happening. But here's the reality. 1/3 of all genetic tests for symptomatic patients are inconclusive due to a VUS. So how many resources should a physician put in to following up on all these VUSs, right? It's a lot of work. In ClinVar, the largest category of variation is VUS or conflicting. So it's a huge challenge to figure out what you should do with VUS in patient reports. Now, you could say, well, the ClinGen expert panels will classify all of them. Well, I already told you early on, there's no way that these expert panels that spend a lot of time on each variant are getting through billions of variants. So we have to have another solution. I will also note that insurers have been denying genomic tests, and some of the anecdotal expressed concerns are these darn VUSs being returned to patients, leading to downstream medical costs in follow-up. And I just gave you a scenario where they should have followed up. So should you follow up on all of them? And so they're all concerned about all these VUSs and the genome and exome sequencing. So I did a little study. I was like, I don't think that's the right way to look at this. So I was like, we can prove this. So I reached out to all my clinical lab friends. We got data from 1.5 million tests from 19 labs across the US and Canada. That data, and then compared the rate of VUS is reported in multi-gene panels compared to exome and genome sequencing. And there is a statistically significant increase in VUS in multi-gene panels compared to exome and genome. And actually, if you look at the size of the panel, it goes up and up as the panels get bigger. That makes perfect sense. But then there's a cliff dropping down to exome, genome. Why is that? Well, it's because of how we actually practice in the clinical lab. We get no phenotype sent to us when physicians order a panel because they've already said, I know what I want. I want this panel for my patient who has this disease. Just tell me what variants, interpret them all. So we interpret all the variants, and what goes on the report, everything VUS and above. That's just how it's done. And we often don't get phenotype to even try to make a correlation. But as panels have gotten bigger and bigger, the specificity of the phenotype for what comes out of that panel is not really strict. So in contrast, in exome and genome, you can't do anything to interpret an exome and genome without phenotype. So we ask for all the detailed phenotype, and you can't report thousands of VUS, so you look very carefully at the variant you're looking at and the evidence and the phenotype, and you only put things on the report that actually have some likelihood of causality. Maybe not high causality, but certainly filtered down from what's going on panels, I can tell you. So that is why this is happening. And I'm delighted to say that after we publish this, there are now insurers starting to cover exome and genome sequencing better. So mission accomplished, although I can tell you there's still a lot of work to do in the insurance coverage area. And two of the laboratories in that study, the lab I used to direct at Mass General Brigham, and Quest, who were in the study, actually had subdivided VUSs. And so we've been doing it for 15 years at Mass General Brigham. Quest has also been doing it for about 15 years. And what you can see is that VUS low category was only ever put on the multi-gene panels, none on exome. It correlates exactly with the argument I was just telling you about practice. So now we've gathered all the labs that are doing subclassifications. We learned Baylor was also doing that, and actually just a few weeks ago learned that LabCorp has been doing this for many years. So there's a fourth group we've just added into this. So this is data we've been aggregating that we're going to publish soon. And what are we looking at? The question of, if you subclassify to VUS low, what's the likelihood it will ever go to pathogenic? And the answer is it has never in 15 years gone to pathogenic with these sub-tiers. So what I'm looking at here is moving up or down from the class it's at. So if you're at VUS low, moving up has only ever gone to VUS mid or high, never to pathogenic. In contrast, those VUS high at the top, the percent that get reclassified, the highest number is that VUS high category. So that's baseline is how many even get reclassified. And then when they do, almost half are going up to likely pathogenic. So what do we need to do? We need to focus our physicians on that category of variation, right? And really do stuff with those, and don't do stuff with the VUS lows. It's a waste of time. So that's my mission now. So I'm co-chairing a new committee from ACMG to put out guidance for how to use subclasses in reporting. It'll correlate and be either before or same time as the new sequence variant classification guidelines come out, so that when we tell labs how to subclassify variants, they then have some guidance on how to use those subclasses in clinical reporting. So that's work in progress. So lastly, I want to just come back to the shared evidence base. So we can do all this work in standardized classification and getting experts involved and all of that, but if we don't have the evidence, we can't do a lot, right? So some of the areas I've worked in is just data sharing. We launched the Matchmaker Exchange now 8 years ago or so to get people to share candidate genes. And it's actually somewhat a brilliant way to deal with people not wanting to share data, because novel gene discovery is a coveted thing. People didn't want to put their candidates out there and get scooped. So we developed a sort of system where you put your candidate in a database. We link all the databases by a common API, and no one discovers your candidate unless they have the same candidate. You're brought together, you both get CC'd on an email, and then you co-publish. It's been one of the most productive data sharing platforms in all of rare disease gene discovery. Great. Okay, I'm not going to talk about that today. I'm going to talk about what we're working on now, which is that works great if 2 people have the same candidate. But what if you have a candidate and you don't match with anyone, which happens a lot. And you're like, I'd just like to know who around the world has the same variant I'm looking at, and I want to know their phenotype. So what we're working on now is what we call one-sided matching. You have your candidate, and you want to put it into a search interface and query every genome in the world. And when you find your variant in somebody, you get back the phenotype. Wouldn't that be brilliant? Every day I get emails, or my team gets emails, saying, I found this variant in Nomad, can you give me the phenotype? And the answer is, no, we don't have phenotype. And they're sad and they go away. So we're trying to solve that problem. I'm sad too. So here's just an example of one group implementing this. The Variant Matcher group, which also supports Gene Matcher, the most widely used platform for gene matching, just opened their database to search all of the genomic data by variant. So here's me. It's very quick to create a username, and you can get access, and you search. So at the very top is me putting in my variant and the phenotype of my patient that I found this variant in. And then what did I get emailed to me? A list of 8 patients that had the variant and the phenotypes of each of those patients, or in the bottom 4, that they were unaffected parents. And quickly, I just ruled out this variant. All the phenotypes were different, or half the people were unaffected, and I can just say, not a candidate, move on. So that's equally useful is ruling out your candidates, putting them in the VUS low or likely benign category, and moving on. But nobody wants to go to 20,000 databases and search them all one at a time and create a username and password for all of them, right? So our goal is to try to link up all of these databases in a federated network like we built with Matchmaker Exchange, but now with true genomic data querying. So these are a lot of the groups that are already sort of coming to our monthly calls working on this. It's actually built off of the framework of the original Global Alliance Beacon framework, but now really focusing on delivering that added phenotype data value. So we're using the Beacon version 2 API to connect all these databases, and we just in the last few months went live with the first node-to-node connection between Variant Matcher and Franklin, which is actually one of the widely used commercial platforms for clinical lab interpretation. And so this is just a little screenshot. So Variant Matcher interface, you can go in, put your variant, and by default at the bottom is you will match against Franklin. And it's just defaulted to checked. So when you do your search of Variant Matcher, you're also going to get back any information from Franklin without you having to go to that database. And in parallel, if you're on the Franklin website, when you search for a variant, you will get a little message. These are two different variants. Variant doesn't appear on Variant Matcher. Great, useful, I don't have to go there. Or variant submitted 726 times, 144 were homozygous on Variant Matcher. That's usually enough for me to say, benign, move on. But if that was like found 3 times, I might go and look that up. And so you can click on the Variant Matcher URL. It takes you over to Variant Matcher. You log in, and you get the phenotype. So brilliant. So the query stats to date since this was launched, Variant Matcher is a much smaller database. 362 queries that went to Franklin. On the reverse is a widely used platform in the commercial industry. So 1,375,000 queries went back to Variant Matcher, leading to 46,000 unique variants matched that span the genomes of 6,800 individuals. So that's just the first node-to-node connection. The power of this system, getting all these databases linked, will be brilliant. I can't wait. OK, so that's a little window into what we're working on. This is a nice slide from Konrad Kaczewski looking at Nomad. And what you're seeing here is the increase in number of individuals from V2 to V4. Unfortunately, that big blue bar is a lot of European individuals added to the database from the UK Biobank. But if you, maybe I can go backwards for a second. If you look carefully at the bar on the right when I click again, that blue bar at the top didn't really change at all. That bar is the number of variants with allele frequency over 0.1%, where we can largely rule out as pathogenic or as benign. I mean, rule out as causal of disease. So what it's saying is we have saturated our understanding of common variation in the European population. We've absolutely not saturated our understanding of common variation in other populations. We desperately need more diversity in Nomad. So we're working on that. We are doing 2 things. So after V4 was launched in the fall, we're going to be launching all of the All of Us data. The fall release that's coming out, we'll be working with that over the coming months to put that in Nomad. That's a very diverse data set. We're also working on the much harder challenge of what I call federated Nomad. So in this case, there's lots of data out there that we cannot bring onto the Broad premises to our cloud platform to process and put in Nomad, because through national laws, the data can't leave those countries. So what we're going to be doing is teaching these groups how to process their data in the Nomad pipeline, create an aggregate allele statistics from that using the Nomad best practices, and then sharing the aggregate data with us that we merge with the Nomad data set so that we can represent diversity without having the underlying raw data. So we're now working with a lot of the countries listed there to incorporate, teach them how to do that. I actually just had a conversation with the European Genome Phenome Archive, where a lot of European and actually even non-European, including H3 Africa and other data, goes. It's like dbGaP of Europe. And they're actually going to set up the pipeline to process data being shared from all these sites that goes into EGA to process it in our Nomad ways and allow that to be incorporated. So that is going to dramatically improve the diversity of Nomad, which I'm very excited about. And with that, I'm going to stop to leave some time for questions. As I mentioned earlier, if you haven't gotten involved in ClinGen, we really welcome the functional community to get involved. I'm also just started a clinical genomics laboratory community to really work together as a community, and you can sign up there. And to have all the URLs of my talk, the slides are publicly available on that link, or you can scan that QR code. I'm happy to take questions.
Stefan Kirov, Flare Therapeutics. Great talk, and by the way, this is music to my ears, the whole federated approach, the walled gardens. I hope this works. So 2 questions. One, going a little bit beyond Mendelian epistasis. People thinking about that or working on that. Essentially, it may, to some extent, alleviate the problem with race and ethnicity. And then one more question around context. So some of these variants are pathogenic in a certain context. Perfect example, sickle cell or cystic fibrosis. So and it's usually infectious disease versus genetic disease. But how do you account for that?
Yeah, it's a great question, and I think one of the challenges is when one lab sees one patient, the statistics aren't there to look for modifiers. But I think, and we're going to have to share the underlying full genomic data so that we can understand what is going on at modifier loci, whether it's within the same gene or it's other genes, other things going on. And in some ways, this variant level matching federated network that I talked about, where we're going to be able to access full genomic data, will actually start to allow more complex queries. And although I didn't talk about it, the Beacon version 2 API is designed that you can do more complex queries. Like, I want to find a case that has both this variant and that variant and this phenotype and things like that. And that mechanism, and one of the queries that we really want that this allows is, do you have any patients with loss of function variants in this gene? Because to build evidence, honestly, it's often not the same variant shared. It's usually a different variant in the same gene, right? So you can start to do, are there any missense variants with Revel scores over 0.8 in this gene in any of those genomes? But also, to your point, what's the genotype at this other locus, and what are the phenotypes of those with this modifier variant or not modifier variant, and start to ask more complex questions like you're getting at, which is the modifiers that influence the penetrance of phenotypes, expressivity, and things like that. So, is this network going to solve all those, questions? It's probably not the most efficient way, just getting all the genomic data together and the phenotypes and really being able to do the traditional approaches that we take. But we will be limited as a global society in sharing data all on, in one server. And so we're going to have to think creatively about how to get access to the entire world's genomic data to answer these kinds of complex questions that require the power of the entire globe to answer them.
Thank you.
Thank you. I wonder if the new ACMG interpretation framework will account for some of the, what I would call an abuse of Richards et al. To the increasing number of often companies who are classifying variants that have as likely pathogenic that have never been seen in a human with the relevant phenotype for that gene. And so, maybe if that scenario is in the framework and maybe it's nearly impossible to get something to likely pathogenic in that framework, then it won't be abused in that way.
Well, while I agree with half of that statement, which is the abuse of the ACMG framework for variant classification, I do not agree that we can't get a variant to pathogenic in the absence of a human observation. And where people go awry are, for example, a nonsense variant in a well-understood exon of a well-understood gene. You absolutely know that nonsense variant is going to disrupt that gene. You don't have to have seen it in a patient in advance. And our framework, unlike the prior one, will allow that variant to be called pathogenic, having never been observed in any human patient. extend that to the functional analysis. If your functional platform is good enough and well validated enough to get to the level of evidence necessary to predict that that variant will be pathogenic when observed in a patient, you can get it. I think the framework, the way we built it, gets you only to likely pathogenic. But I would argue, if we consider likely pathogenic 90% likelihood of pathogenicity, absolutely a lot of these good functional assays, not the ones that are like, we built this functional assay, we validate with one variant and it's great. No, like the good ones that have been really well validated, they can get you to that level, and I would argue in some cases could get you to the 99% level even. Now, with the framework we have sort of cautioned, and we are maxing out at 8 points on the functional data so that you can't get above likely pathogenic. So we have built a little kind of cushion there for conservativeness. But that's not to say, that's kind of to prevent overinterpretation. The really good functional biologists that working with the really good clinicians who can ensure that that functional assay really recapitulates human disease, I believe you can get to a 99% confidence. I don't know how many, actually, how many people agree with me? Raise your hand. Okay, how many people disagree with me? All right, well, this audience speaks. Okay, did that help answer your question, Fritz? Or do you disagree? You didn't raise your hand.
I agree, actually, so I maybe misstated the question too strongly, but I guess the main question is does the system account for cases where there is no phenotype observed person with it?
So yes, like human observations are actually a really key component. Both human observations without phenotype. Now, penetrance has to be considered, age, like all sorts of factors, and how you think about the presence of a variant in a patient and do they have the phenotype? How specific is that phenotype? So the number of points you can apply varies depending on the specificity of the phenotype. There's all sorts of things there in how we think about human observations, as well as when you're talking about recessive, the in-trans observation of a pathogenic variant, all sorts of things. Have you looked at all of the genes implicated in that disease and ruled out variation in others? All those things are built into the sort of human observation side of it. So yes, we definitely are working on that.
All right, we're going to cut it we have 4 questions remaining. So Sven, then Moaz, then Doug, then Hannah.
Yeah, thank you. Sven Diederichs, German Cancer Consortium. Very interesting, great overview. I would like to delve a little more into the comparison between functional assays versus predictions. I was a little surprised to see that I get 4 points for a cellular assay, a functional assay for the individual mutation, and also for a Revels score, which is, and my question basically is what's the rationale for having these equally weighed? And is this also something that from your clinical routine you would say, in my personal professional experience, predictions are as good as functional assays?
Yeah, I will admit we have been struggling with this question because in my mind, the two are overlapping. And if you have a perfect predictor or a perfect functional assay, in theory, you shouldn't be adding the other on top of it, right? But none of them are perfect, and each does contribute distinct information. So we do feel that you need to bring them together and combine them. But we do then cap that section of the framework to try to reduce the impact of the double counting concept. Have we gotten it perfect? Probably not. And this is where, 4.1, let's get something out there because it will be better than what we have today. And then we will continue to iterate on that. And we sort of welcome this community, certainly, to think about how to think about that. That said, the 4 points for a Revels score that high, very few variants. And actually, Anne sitting in the audience, she and others in the ClinGen group did an analysis of genomes. That paper is on preprint, I think, right now. On average, all of us have, I think it's 1 variant that hits that point. So there are very few variants that hit 4 points. So it is definitely an area of anxiety of a lot of people, of like, should in silico predictions get that high? We are going to be piloting this framework. There's actually 90 labs signed up to pilot. We're going to see how this goes with the framework we can develop. But I do imagine trying to slice and dice the functional versus in silico is an area that needs to really be tested and figured out. And we may not get it perfect, but we will continue to work on it and welcome the community's input on that. Yeah, it's a good question.
Mo David, Baylor College of Medicine. Thanks for such an amazing talk, Dr. Rehm. I had a question kind of about the gene curation expert panel, variant creation expert panels, and the different lines of evidence. And so a lot of the protocols, kind of like you showed, they've been updated version by version, but they still rely fundamentally on some really classic lines of evidence. And for this community especially, the way the functional assays are approached here, there's a lot of local context built into the assay. So you like saturate an entire locus, for example. So one of the things that I've been thinking about is for a gene disease assertion, for example, if you had a candidate variant, and that candidate's like in the middle of the saturated locus, you also provide a lot of local contextual experimental measurements in addition to just the candidate's experimental measurement. And so I was wondering, do you think that is a certain level of evidence that is kind of separate from some of these current lines of evidence? And if so, would that be something that's maybe like a MAVE-specific line of evidence that gets built into some of these, some of the protocols and the thinking?
Yeah, just to make sure I followed your question, I think there's a couple of things embedded in there. One, we think differently about patient observation of functional, if you do, let's say, an alpha-galactosidase enzyme assay on a patient because they had a GLA variant for Fabry disease, and you find that the enzyme activity levels are decreased in that patient. You've implicated the locus, but you actually can't say for sure that it's the variant you've observed versus some deep intronic variant that's actually leading to altered splicing and so on. So like how we think about patient data derived from the patient who has the entire context of the genome around that variant, right? And you may be looking at the wrong variant. So that's one challenge. Now, you were more talking about the assay itself, which has its own biological context and in vitro or in vivo, depending. That is where really understanding how well that system tests, all functions of the protein. Does it just test some or does it test some? How is that environment different? And we certainly have pseudodeficiency alleles that highlight the fact that there are differences there. But it will be the test of those assays and how they correlate with other measures of pathogenicity that don't rely on the functional assay to really try to suss out the different contextual aspects of those functional assays. I don't know if I totally answered your question, but.
Yeah, I think so. It's just, I think if you had like a variant X, right, and then you had every variant plus or minus 100 bases as well. So I just, like, when I look at data, I just feel like that's a different thing I'm looking at than the classic, we're, looking at a paper that's looked at 2 or 3 candidate variants and like a small smattering of negative control variants.
It's more the mutational scanning approach of looking
Right.
at, absolutely, and I think you will learn more about that system, having systematically altered variation across it to understand the system better. So I definitely think that is a better approach than randomly picking a few pathogenic, which is actually why, one of the reasons why really a more thoughtful approach can get you more points in that function versus I had at least 10, pathogenic or benign that we only limit to like 2 points sort of thing. So I think there's more there, and we certainly welcome the input to how to think about this, in deeper ways, but absolutely it's a question.
Thank you.
Anna Gloin, Stanford. That was fantastic. I was really interested in what you had to say around the VUS interpretation in exomes versus the gene panels.
Yeah.
And I guess I wanted to know your opinion about the revamping of gene panels. There are a lot of historic members on those panels that we now know have nothing to do with particular conditions, yet they remain on those panels. Is there a time to revamp them and to get rid of some of those legacy genes that have been shown now to be not related to the disorders.
Yes, and sometimes I'll put in slides that, so in Vitae, the moment those expert panels got published on Brugada syndrome, their panel went from 21 genes to 1. Now they did have a way that you could opt to add in, if you knew what you're doing, preliminary evidence genes, and that dealt with the fact that there is a large proportion of the physician population that thinks that bigger is better. So when you're a clinical lab dealing with the marketing constraints of the orderers, they didn't want to be like, you can't get, GeneDx has the bigger panel than Invitae. So there's all sorts of market forces that are here, but the labs are trying to do the right thing. And so the default ordering panel for Brugada syndrome at Invitae became 1 gene overnight when that paper was published. Just released in the last week from the Cancer Institute or professional guidelines is a new recommended panel for cancer screening that is absolutely built on the ClinGen review of all the cancer genes. So this is happening. It is informing what gets put in panels. We work through the Global Alliance in the National Initiatives Forum across many different countries and clinical lab networks. Genomics England and Australian Genomics have their panel apps. That is evidence-based review that's being used to inform those panels, and they're participants and submitters of GenCC. So we as a community are all kind of working together to really inform these genes, agree on which ones are valid, and get the other ones out of there, basically, yes. So slow, but it's going.